We can do a very simple association study. And this is also to give you also example of our own work. What you basically, because up till now we have spoken about association studies as if we are looking to two different genetic markers. Of course, what you want to do is use this principle to identify disease genes, and you can do that by seeing whether there is a association between your disease gene or one of your disease genes and a particular marker on a chromosome. So then, if you know that your disease gene, there is association, or your disease is associated with a particular genetic marker, then again you know that there is a gene close to that marker. So that's again the principle that you use in association studies. But you have to think that the second marker that we're looking at is not a particular genetic marker, but is your gene of interest, is your disease gene, or the allele that is important in the development of disease. So let's just try to explain that with one example. So a very simple study for association could be if you want to find out whether a particular genetic marker, a SNP, where a particular SNP on a particular chromosome is associated with your disease. So you just take diseased individuals and healthy individuals, and you just look to the frequency of that particular allele in your patient group and in your healthy controls. And here you can see the SNP allele 1 is in patients it's 20% frequency, in healthy controls it's 80% frequency. This is a very atypical example, but you already can feel that there is something happening with this genetic marker because you see it less on this particular allele, because you see it less in patients than in controls. So here this gene, important in development of this disease, is associated with your SNP. And if you know that SNP is, let's say, on chromosome 1, a particular region of chromosome 1, then you probably know that there is a gene important for this disease on chromosome 1. So this is the way you can go for these SNPs. Of course, you will not look to one SNP, but you will again screen all the SNPs on all the chromosomes. You do a kind of genome analysis with all the SNPs available. You can do that today with all this DNA chip technology. And in this way, you look to find SNPs that are associated with your genes. Again, one very simple example from the real world. Some years ago, we have looked at this gene, which is called leukemia inhibitory factor. It's a protein that plays a role in the immune system, and there was a question, could that gene be involved in development of multiple sclerosis? So we looked at that gene and we found that there was one polymorphism in the gene. Actually were two, but this is just one of the examples here. On this position in the gene, some individuals had a C and other individuals had a T. So this was our polymorphism. It's a SNP. You have C or T. One allele is C, the other allele is T. How can you make that visible in an individual? How can you make that visible in the DNA of that individual? Well, you just take DNA, you do an amplification with PCR of this region, and you cut this with this restriction enzyme. And this restriction enzyme will cut when the particular sequence is there. So in that case, you will have allele C, because if the fragment can be cut, the sequence is there and you have allele C. If the fragment cannot be cut, then this sequence will not be there, so you have the other allele. So you do a PCR of this region, you cut with a restriction enzyme, you separate the fragments on a gel, and you can read the genotype of those individuals. This one is a CC, this one is a TT, and this one has the 3 fragments, so both the cut fragment and the non-cut fragment, so this is a CT. So with this simple analysis, you can study the genotype of individuals, and you do that in patients and controls. And that's what we did. We looked at healthy controls, MS patients, and also rheumatoid arthritis patients, which we often use as controls. And we looked to the frequency of, this should be allele here, of the alleles of one of the SNPs and the alleles of another SNP, and we looked at the frequency of these alleles in healthy controls, MS patients and rheumatoid arthritis group, and we did the same for the genotypes. But to cut a long story short, we did not find any difference, indicating that there was no association, there was no linkage disequilibrium between this SNP and development of disease. So we could rule out the possibility that this gene was important in development of this disease. So in this case, we looked only to one SNP. Like I said before, today we look to thousands or millions of these SNPs. Okay, so now you know a little bit how these genetic technologies are applied to identify the genes. Let's see what they have learned so far. Well, the most frustrating part in genetics and autoimmune diseases is that one of the genes that is really important is known for already 40 or 50 years, and these are the MHC genes. The MHC genes, you know, in humans these are the HLA molecules which are present on the outside of our cells and play an important role in the immune system. And it appears that for most of these autoimmune diseases, again, you can read the insulin-dependent diabetes or type 1 diabetes, MS, for instance, to make sure there is a huge difference between type 1 and type 2. Type 1 is considered to be autoimmune, type 2 is not autoimmune. And here you see multiple sclerosis, rheumatoid arthritis, some other diseases. And you can read here that they have all an association with one of these HLA molecules. It can be HLA molecules of the class 1, let's just be 27, you know, HLA A, B, C, that's class 1, DR, DP, DQ, that's class 2. So you see for some we have association with class 1, for others we have association with class 2, MS for instance is associated with HLA DR2. And you can read here the relative risk, meaning that if you carry this gene, what is your risk of developing disease as compared to individuals who do not carry that gene. And for ankylosing spondylitis, there is a clear genetic risk because individuals that carry HLA B27, they have a 90% increased risk of developing that disease as compared to individuals who do not carry that disease. Ankylosing spondylitis is a chronic inflammation of the spine. It's a kind of rheumatic disease, but this clearly is associated with HLA B27. And for instance, MS is, like I said, correlated with HLA DR2. And here the risk is lower. This table is 5. Sometimes you see differences, but meaning that patients who do not carry that gene, individuals who carry that gene have a 5 times higher risk of developing MS than those who do not carry. So meaning that it clearly plays a role in disease, and it's a very important genetic marker, but it cannot be used, for instance, for diagnostic purposes, or really to find out who will develop that disease. Therefore, the importance is not high enough. This marker is sometimes used in the clinic, but I think this is the only example. All the other genetic associations are not used in the clinic to, for instance, screen or diagnose patients, so they're only useful for the science, but not for the clinic, because their effect is so small. It's significant, but it's small. The problem with the MHC is that this is a cluster of genes with many proteins that are important in the immune system. You see a lot of genes there, and a lot of these genes are also in linkage disequilibrium, meaning that this is a region where you have these associations between the genes that are present, and therefore it's sometimes hard to find out which is the true gene that is associated with your disease, because all these genes are associated. So if this one is associated with your disease, the other one is probably also associated, and the other one is also associated. You never know which is the true one that is important. And in some cases it was shown that class 2 genes could, for instance, play a role in MS, and some studies have also shown that some class 1 genes appear to be protective to MS. So it's a very complicated story, but today we know that class 2 in MS and also rheumatoid arthritis and other diseases is important. We know that since, I think, 1960s, when all these sophisticated genetic technologies were not yet there. We still don't know why actually patients or individuals with these MHC types have a higher susceptibility of developing MS or arthritis or whatever. Again, you see a lot of hypotheses in the literature, but we still don't know. One of the simple explanations could be, of course, that individuals who carry, let's say, HLA-DR2, that they have a better way to present specific antigens to the immune system. And maybe they present the antigens that are important in developing this autoimmune cascade of events. So it's possible that they can do the better job in presenting these autoantigens, but this is still not completely clear. So despite the fact we know this, we cannot use it in the clinic, and we still don't know really what is the function in this association. Another problem, if you would go over the literature of the last, let's say, 20 years to look at those association studies in MS, but also in other fields, there was a huge problem in confirmation. So some studies came up with an interesting gene, and then others could not confirm those data. One of the problems was that you need also large sample sizes. So as you've seen in one of those previous publications that we published in 2005, I think, we used 100 patients. Today it's really impossible to publish with 100 patients such a story. You need way larger groups of patients and controls to look for genetic association, because you may look for genes with low penetrance, so meaning that those genes have only a little role in the disease. Despite all that I said, in the early stages of MS genetics, people have really approached the way to identify genes by using the linkage studies, by using actually small families. The problem was we did have the large pedigrees, but sometimes you have two MS patients in one family, or maybe if you're lucky 3 MS patients, and people were able to identify quite a lot of those small families and try to use all these linkage studies to identify genes for MS. And actually there were, I think, over 11 important studies published from 1996 to 2004, usually with this multiplex family, so 1 or 2 patients in small families, using macrosatellites, so the STRs, the polymorphisms where you have this length variation. And as you can see, the frustrating result of those studies, many published in top journals like Nature and Science, they could only confirm the genetic association with the MHC region. So every study came up with MHC and then a list of other genes. There came another study, MHC and another list, MHC another list. So there was a very difficult time to find out what is really important in those studies. After 2005, you saw that people were using DNA chips, and so they went from, let's say, the macrosatellites, the STRs, to the SNPs, because technology became available to do a wide variety of SNP analysis in these patients. And you can see the numbers of individuals and families that were included in those studies. This publication of 2005 showed again chromosome 6. Well, if you can read it, there's a nice log score. Chromosome 6 is MHC. This is what we knew for many years. And then you saw that you can read the other profiles here, and I think it's also this table. This study showed you have a log score of 11.64 6p21. So there's chromosome 6, region p21. You have a log score of 11. I told you that if you have a log score above 3, then you're sure that there is linkage. So here is linkage, but it's HLA molecules, so that's known for 40 years already. And the other genes that came out, you see that they had, the researchers identified the region where there was linkage, but basically have a log score below 3. So you don't know what to do with it. Is it a true linkage, or is that just by chance? This research went on like this for some years. And then in 2007, there was some important papers, also published in the high-ranked journals, Nature and the New England Journal, showing that they found a new gene for MS. And you could also see that in the newspapers at that time, a gene for MS identified. It was an IL-7 receptor. Now, at that time, they also demonstrated that IL-2 receptor was important. And we as immunologists, we like this a lot because it showed that the immune system is important in MS, because IL-2 receptor, of course, is a gene that is important for the proliferation of T cells. By the way, it's also important for regulatory function of the immune system, so that could also be interesting. IL-7 is, or IL-7 receptor is a gene, of course, that binds to IL-7. IL-7 is a cytokine that's very important in keeping your immune system in balance, do the homeostasis of your immune system, so it helps to keep the number of cells in your circulation in balance. So this could also be very important and very relevant for MS. But then if you look to the data, and you cannot read that, I understand, but you should always read all these papers to find out what's really the significance, because people said, wow, we have these new genes, but what do they do and how important are they? And then you have to look also to the odd ratios, and well, I will read it for you. And you will see here for 6p21 an odd ratio of about 2, meaning this is again MHC, of course. So here it's a very high odds ratio, meaning that there is a good chance of probability that this gene indeed adds to your susceptibility for the disease. But the odds ratios for the other genes, and you see here, I think this is the 2, IL-7, is 1.25, 1.18. So meaning it's very little above 1, meaning that it adds very little to the genetic susceptibility. So these tables, they show you there is a significant association, but it probably only has a tiny influence on the genetic makeup. It only increases, if you carry this gene, it will only increase your chance from 1 to 1.25. So which meaning that it has a very low added value to the genetic component of the disease. It is important, it is significant, but you cannot do much with this. You cannot use it for clinics, you cannot use it for diagnostics. The only thing that's important for us is indeed it's an immune gene, and for instance our group started also to explore these SNPs in patients to find out if these SNPs or patients carrying this particular alleles have a different immunophenotype. This work went on in 2000, so last year. There was another important publication, and you see that you don't find individual authors of these publications. You have this, the International Multiple Sclerosis Genetic Consortium and the Wellcome Trust Case Control Consortium number 2. So usually this is a group of more than 300 people that are working on this. Some have really contributed in genetic analysis, others have sent the samples for these studies because you need a lot of samples. As you can see, they add up almost to 10,000 cases that were studied in this particular study. It's a GWAS, it's a genome-wide association study, so all the SNPs that are available were studied. And what came out of the study, again, HLA, also the IL-7 receptor, IL-2 receptor, and a lot of immunologically relevant genes were overrepresented. Among these mapping close to the identified loci, and particularly implicate T helper cell differentiation in the pathogenesis of multiple sclerosis. So again, it showed or it confirmed some people that MS really is an immune disease, but that of course we already knew. Therefore, we don't need these studies of 10,000 individuals. Some interesting genes were picked up, and you have to also be careful by reading the last sentence. It says immunological relevant genes are significantly overrepresented among those mapping close to the identified loci. So basically what they do is you find a SNP which is linked to your disease, and then you look if there are genes close to that SNP. And you have to be careful not to bias there, because some people will say, okay, there is an interesting gene here, but there is also an interesting gene there. This may be an immune-related disease, so that is probably interesting because it's related to MS. And then they say, okay, this is probably associated gene, but it's only close to the SNP. So I always have to be careful. That's not always the right gene to look at. It's a guess. And this is seen, and on this complicated slides, again, you're unable to see that, but basically you see the whole genome, coming from 1 to chromosome 22. What you see here is dots that go like this. Usually they put it like this, and they call it Manhattan plots. And you have all the chromosomes, and when the dots go higher, then there is a good chance that these are all SNPs. Every dot is a SNP. And you see that there is a high chance for linkage for this region with these SNPs, high chance for linkage with these regions. So this way you can identify, here you see a lot of SNPs there. This way you can identify the SNPs that are correlated with disease. So this is again the story for MS, and you see a lot of SNPs which show some kind of correlation with odd ratios, usually not going above 1.3, so indicating that each of these SNPs, each of these genes only has a little contribution to the genetic makeup of the story, the genetic makeup of these patients. And then they identified some candidate genes which are close to those SNPs. Again, be careful, it's not always that this gene is one-on-one related to the SNP. There can be other genes around. So these are candidate genes, and some of these candidate genes appear to play a role in the immune system. What is also interesting from this slide that you cannot read is that if you could read it, you will see here other names of other diseases, such as rheumatoid arthritis, type 1 diabetes, and what you see is that some SNPs that are seen for multiple sclerosis are also identified as candidate susceptibility factors for other autoimmune diseases. And this is also something that we as immunologists like a lot, because it indicates that there is a common ground in some of these diseases. So the main question, and this is more or less where we are today, we have a long, huge list of candidate genes. Like I said, 1 or 2 which are really important, but the rest probably has only very little role in disease. And the question, of course, is what can we do with that information? Like I said, we cannot use it in the clinic, so no one is using this information for diagnostic, even not for prognostic, because it would also be interesting to find out, based on your genetic makeup, which patient would develop much more aggressive disease, other patients may develop a more mild disease, especially in MS. The course of disease can vary a lot between patients, but we have no information yet about which genes are important in aggressive versus mild disease. Of course, these kind of studies will come in years to come, but today we cannot use it for this. We try to use it to find relationships between molecular pathways and disease mechanisms. So again, if genes come out of these studies, it's interesting to find out if these genes could indeed play a role in one of the pathways or the mechanisms involved in disease, and this could bring new information that also helps us to identify targets for therapy. And of course, another way to use that information, and that's probably the most useful in the near future, is to use those SNPs to stratify patients and identify which patients may be better off with a particular treatment. Today, all the treatments that are developed also for MS are given to all MS patients. But we know that based also on the way they progress, but also on the phenotype they have, that there probably are differences between MS patients. And these genetic studies can help us to discriminate groups of MS patients, and if we do this type of analysis in the same framework as the development of a new drug, and today this is the way most pharmaceutical industries work, genetic comes together with the clinical studies, then of course the outcome of these clinical studies may also give us SNPs that are related to a better clinical efficacy for a particular drug. So we'll see that those genetic analysis or those SNPs may also be used for what is called personalized, or what is better probably called stratified medicine in the future. And probably some of you may know 23andMe, which is a company in the US that today will perform genetic analysis, also genotype analysis for every individual. So what you can do is just go to the website, order this kit. You take some of your mouth fluid and put it in a small box, send it by FedEx, and they will do your genetic analysis. And sometimes it's interesting to go to those websites, because initially what you could read of what kind of analysis that they were doing was not so much scientific up to date, but you see that they're using scientific literature to bring in all the SNPs, for instance, that have been identified for rheumatoid arthritis or multiple sclerosis. And what you can do is send your DNA and do the analysis for these disease-related SNPs. Of course, this is still not so very useful. Then they claim, for instance, that you can screen for susceptibility factors for type 2 diabetes. And what you can do is then determine your genetic risk factor, your genetic risk for, in this case, development of type 2 diabetes. You can do that for many, many different diseases. The list is pretty long. Question is, of course, what can you do with this type of information? Sometimes, of course, you can use a particular diet or do kind of exercises to reduce your chances of development of these diseases. You can also screen for inheritable conditions, even diseases like cystic fibrosis or types of breast cancers, and then you can send your DNA and ask whether or not you carry those genes. In Belgium, these type of analyses are always done in genetic centers. There's of course special legislation around this, but people now can escape this part and just send their DNA to the US and find this information. What's probably very useful is the correlation, like I said, with drug therapy. So one of the tests they do is also to determine your sensitivity for warfarin. It's actually a drug that is used as a blood thinning drug for patients, for instance, with a chance of developing stroke. But they saw that in a subgroup of patients this therapy led to a lot of brain bleedings, and that's why they found that actually based on your genetic makeup, some people have a higher chance of developing these bleedings, and that's why with this genetic analysis you can discriminate, or you can identify individuals which should use a lower dose of this drug in order to not have this unwanted side effects. And it's interesting to read the slide because the world is going in another direction. Usually you go to your doctor and ask him, what should I do? With this test, you go to your doctor and you tell your doctor what to do. So this is opening up, I think, a completely new world, and it's interesting for you to follow that. Like I said, I was wondering when I was making these slides if I should tell you all about these all genetic studies, because you know today that you can do the whole genome sequencing. I think it's already less than $1,000 to do whole genome. And you see here the price of whole genome sequencing. This is this curve, I think, the cost of genome sequencing. You see the number of genomes that have been sequenced already. So today we are somewhere here. You see more than 10,000 individuals have been fully sequenced, and this will rapidly increase. So you will be working in a couple of years in a situation where most of the genome sequences are available, and of course you probably will use other technologies to look for genes, identify mutations, and so on. Although some of the things that I probably told you today will still be useful then. But this will of course completely change the world in genetics, also genetic drugs and so on. So it's important that you know all about these genetic technologies. I'm sure there will also be somewhere in one of the courses to come. By coincidence, this week we are organizing, together with other universities, a whole course for PhD students on these so-called next generation sequencing technologies, which can sequence whole genomes or whole exomes, so all the sequencing, all the exome information that is in the genome. So this will open up new possibilities. Another project that is really important to end with is a 1,000 genomes project. So after the genome, human genome project, where basically one sequence was determined for the human genome, there is a project ongoing now where at least 1,000 individuals will be sequenced. This information will become available also in publicly available libraries. And this, of course, will give us way more information about the genetic variation, genetic diversity, and all of this information, of course, will be very useful in years to come also for the genetic studies that we are doing. So this, to end up with, is some literature that can be used to read. I put on some new, if you're interested also in the new evolution, then I don't think this is, you have to read it, but it can be seen as examples of publications where you will also read a little bit more about the next generation sequencing, also how it is used, for instance, in cancer, because there it's becoming very important. And finally, I hope that I have been able to convince you that these complex diseases that have both genes and environment is important. Sometimes if you study the rare diseases, it can learn you a lot about the more complex diseases. The positional cloning has provided us all the genes that are important in single gene disorders. But to find the genes in more complex diseases, you need other technologies such as the association studies involving, for instance, the SNPs, the SNP maps, and well, you need large numbers. Both numbers of samples, but also large collaborations of researchers to identify those genes. And you have to follow up the new technologies because they will open up many, many new possibilities. Okay, and last, which is maybe the most frustrating part. So far, many of those genetic studies have not been able to be translated really into clinical new drug development. But I'm still optimistic, and I think that some of those approaches will still help us to identify also new drugs. So that's also the focus for the coming years. Thank you.